Good morning, students. Today we are discussed about functions of ethylene hormone. Earlier we discussed about the structure and the biochemical structure of ethylene hormone, and today we discussed about their role or functions in plants. Ethylene is capable of inducing adventitious root formation in leaves, stems, flower, stems, and even other roots. Vegetative stem cuttings from tomato and petunia make many adventitious roots in response to applied oxygen. But in ethylene, in sensitive mutants, oxygen has little or no effect, indicating that the promotive effect of oxygen on adventitious rooting is mediated by ethylene. Ethylene also plays a role in the morphogenesis of crown gall tissue and is a negative regulator of root nodule formation in legumes. Ethylene has also been shown to act as a positive regulator of root hair formation in several species. This regulation has been best studied in Arabidopsis, in which root hairs normally are located in the epidermal cells that overlie a junction between the underlying cortical cells. In ethylene treated root cells not overlying a cortical cell junction differentiate into hair cells and produce root hairs in abnormal locations. Seedlings grown in the presence of ethylene inhibitors such as silverines as well as ethylene insensitive mutants display a reduction in root hair formation. These observations suggest that ethylene acts as a positive regulator in the differentiation of root. Ethylene regulates flowering and sex determination in some species. Although ethylene inhibits flowering in many species, it is induced flowering in pineapple and its relatives, and it is used commercially for synchronization of pineapple fruit set. Flowering of other species such as mango is also initiated of ethylene on plants that have separate male and female flowers. Ethylene may change the sex of developing flowers. The promotion of female flower formation in cucumber is one example of this effect. Recently, a gene responsible for andromonaceae, plant carrying both male and bisexual flower, in melons was identified as encoding an ACC synthase, a mutation that reduces the activity of this ACC synthase genes result in the formation of the bisexual flowers in these andromonaceous lines. Ethylene promotes growth of submerged aquatic species. Although usually thought of as an inhibitor of stem elongation, ethylene is able to promote stem and petiole elongation in various submerged or partially submerged aquatic plants. These include the digots, Ranunculus scleratus, Nymphoides peltata, and Calitric platycarpa, and fern Reginaldium diphyllum. Another agriculturally important example is a deep water rice, Oryzis sativa, a cereal. In these species, submergence induces rapid internode or petiole elongation, which allows the leaves or upper parts of the shoot to remain above water. Treatment with ethylene mimics the effect of submergence. Growth is stimulated in the submerged plants because ethylene builds up in the tissues in the absence of O2. Ethylene synthesis is diminished, but the loss of ethylene by diffusion is retarded underwater. Sufficient oxygen for growth and ethylene synthesis is the underwater parts is usually provided by parenchyma tissue. Ethylene stimulates internode elongation in deep water ice by increasing the amount of and the sensitivity to gibberellin in the cells of the intercalary meristem. The increased sensitivity to gibberellic acid in these cells in response to ethylene is brought about by a decrease in the level of abscisic acid, a potent antagonist of gibberellic acid. Ethylene enhances the rate of leaf senescence. Senescence is a genetically programmed developmental process that affects all tissues of the plant. Research has provided several lines of physiological evidence that support roles for ethylene and cytokinase in the control of leaf senescence. Exogenous applications of ethylenes or ACC accelerate leaf senescence, and treatment with exogenous cytokinin delays leaf senescence. Enhanced ethylene production is associated with chlorophyll loss and color fading, which are characteristic features of leaf and flower senescence. An inverse correlation has been found between cytokinin levels in leaves and the onset of senescence. Ethylene acts on the abscissin layer. The shedding of leaves, fruits, flowers, and other parts organs is termed abscissin. Abscissin takes place in specific layers of cells called abscissin layers, which become morphologically and biochemically differentiated during organ development. Weakening of the cell walls all at the abscissin layer depend on cell wall degrading enzymes such as cellulose and polygalactosuronase. Here is the diagram. Formation of abscissin layer of jewelweed. First, during leaf abscission, 2 or 3 rows of cells in the abscissin zone undergoes cell wall breakdown because of an increase in cell wall hydrolyzing enzyme. And in the next diagram, the protoplast released from the restraint of their cell walls expand the push apart the xylem trachea cells, facilitating the separation of the leaf from the stem. And another function of ethylene, it breaks the seed and bud dormancy in some species. Seeds that fail to germinate under normal conditions are said to be dormant. Ethylene has the ability to break dormancy and initiate germination in certain seeds such as cereals, in addition to its effect on dormancy. Ethylene increases the rate of seed germination of several species in peanuts. Ethylene production and seed germination are closely correlated. Ethylene can also break bud dormancy, and ethylene treatment is sometimes used to promote bud sprouting in potato and other tubers. Ethylene also promotes the elongation of growth of submerged. It also promotes the lateral cell expansion. It also at a ethylene changes the growth pattern of seedlings by reducing the rate of elongation and increasing lateral expansion, leading to swelling of the hypocotyle or the epicotyle. In dicots, this swelling is a part of triple response, and ethylene induces the lateral cell expansions in plant. So it's finished. Thank you, students.